Hello lovelies, in this video the brilliant Dr. Redbirds is going to be looking at one of your practicals for your A-level biology. So we're going to be looking at chromatography and we're going to be relating it to the pigments in leaves. Now we're going to show you how to do this practical, and then when we get to the end we're going to show you how to interpret the results as well, because this is the key thing that you would need for your exam.
So today we're going to look at using chromatography to separate photosynthetic pigments, which is one of the practicals that we have to know. And just a reminder that photosynthetic pigments are present in photosystems that are found in chloroplasts. And remember that different plants will have different concentrations of the different pigments in their chloroplasts, and that gives them their different colours, the different colours of their leaves, and it allows them to absorb different wavelengths of light. This gives some plants adaptations, so they could be tolerant to shade, so not very high light intensities, or different light colours, so light pass through the leaves, or high UV light, for example. So one of these examples is Tradescantia, and we're going to look at these leaves today. They have very red or purple pigments in their leaves on the underside, which you can hopefully see here. They are caused by photosynthetic pigment group called anthocyanins. These actually act as a UV protection for the plant. So because they absorb more on that light intensity spectrum, they can actually help the plant survive in quite high light intensities. And so remember, we've got this range of wavelengths that light can be absorbed in, and the photosynthetic rate is also dependent on which light wavelengths they can absorb, which is why they need this range of pigments. So the idea behind this practical is to test one of our ways of separating mixtures, and chromatography is one of those methods we can use, and it separates out molecules from a mixture. The molecules are dissolved and then they move through what we call the mobile phase, which is normally a liquid solvent, and they will get fixed onto a stationary phase, and they will spend different amounts of time in the mobile or the stationary phase, which means that they get separated out along the piece of paper or plate that we're using over time. And there'll be different reasons why they separate at those different rates. Okay, so here we have a diagram about what we've just mentioned. So we have our mobile phase, which is the liquid solvent at the bottom, and it's just touching that stationary phase, which is either a solid piece of paper or a thin layer chromatography plate. And that liquid is going to be drawn up, it's going to move up over the stationary phase, which is why it's the mobile phase, because it moves. And then that's going to ultimately drop off at different stages the different pigments and separate them out from the one group of pigments we had at the start. We seal it all together and we make sure we have lids on everything that we're doing with this, because the solvents that we're using are obviously quite volatile and they can evaporate quite quickly. Partly we don't want them to evaporate, and also we don't want too many of those chemicals in the air. Okay, so the rates of migration, so how far and fast the pigments are going to move, is going to depend on three things. How soluble they are in the solvent, so different solvents will pick up and dissolve different molecules in different amounts. Their affinity to the stationary phase, so how well they interact or bind to the stationary phase. If they're really strongly interacting with it, they won't move as far. And their mass, so the smaller the molecules are, the further they'll travel. This is less important, but it's partly something you should know as a way that we can separate molecules by their mass as well. So the distance that the substance has moved through the stationary phase relative to the distance that the solvent moved can be calculated and give an RF value. So we can actually quantify the movement of these pigments, and that can help us to identify them, and it can help us to compare them as well. So if the solvent moved all the way to here, this would be our distance travelled by solvent. And you can literally measure that in millimetres. And then for each individual spot, we can measure the distance that they travelled from the baseline. And so that will give each individual distance of the travelled by the substance, and then you can use this formula to calculate an RF value for them. So each photosynthetic pigment in this case will have a specific RF value when run under those conditions. So these can be compared to their known RF values in a database to try and identify the pigment. However, as I said, it's under those certain conditions that that RF value is produced. So more than one substance can have the same RF value for a particular solvent, and whether you use chromatography paper or thin layer chromatography or whatever the stationary phase was. So the different mix of solvents and the different stationary phase can have an effect on the RF value. So you need to be able to compare it to known values that were run in the same way. So using the same sheet and using the same solvent. And sometimes you might still get conflicting numbers. So you might need to run different chromatography sheets and different solvents to try and identify all of the different pigments separately by their RF value. But we can also, in terms of photosynthetic pigment, for example, as well, we can also use their colour to help us. So if it has an RF value within a certain range and it is that colour, it's going to be more likely to be a certain pigment than if it was in a similar range but it was a completely different colour. That can help us separate pigments that have similar RF values as well. Okay, so for our equipment, we're going to need some capillary tubes, we're going to need some filter paper or TLC plates, either is fine. 2 types of leaves, and you could have leaves that you know are adapted to sun or shade, or like we've got here 2 different coloured leaves, just that you've got something to compare. A pestle and mortar, some glass tubes or test tubes and bungs, running solvent, extraction solvent, these can be different, a ruler, a pencil and some drawing pins. And all of my equipment is set up here in the image. So our first step is going to be tearing up our plant leaves into small pieces, and I'm going to show you how to do that in the video, and then place it in a mortar and pestle, and we're going to grind it with a small amount of our extraction solvent. Then we're going to get our, in this case, filter paper, draw a pencil line about 1 centimetre from the base, and use the pencil to mark that start position. Remember, we have to use pencil because if we used ink or anything from a pen, then that's going to run up along with our pigments and that's going to disrupt our results. We need to repeatedly spot, using our capillary tube, a small quantity of the leaf extract onto the centre of that line, allowing the spot to dry between each application. It's really important to allow that concentration to build up to make sure it's dry in between, also to make sure it's very dry before you put it into your chromatography. Suspend the paper or the plate in the glass vial with the lid on, so with the bung on, and have your solvent in the bottom. Ensure that the chromatography plate dips into the solvent but that the spot of the leaf extract remains above the surface of the solvent. Otherwise, then it's just going to dissolve into the solvent at the bottom and it's not going to run up and carry up in a nice neat line, which is what we want. Allowing the solvent to run up the chromatography plate until the solvent front is near the top. So there is no time limit here. Different solvents will take different times to run up. You just need to stop it until when the solvent front, so where you can see the solvent moving up the paper, gets to near the top, because you don't want it to run off the edge, because you need it to be able to calculate your RF values. So as soon as you take it out, you draw a pencil line across the top to mark the solvent front. And you need to do this quite quickly because the solvent will evaporate and dry quickly, so you won't be able to see it then. So you need to do it as soon as you take it out of the tube. And then we're going to be able to use our chromatograms to calculate the RF value. One thing I will say here, obviously I'm doing it in a video, but take photos of them as well straight away with a ruler next to the picture, next to your chromatogram, and in good lighting, because the photosynthetic pigment colours are actually affected by UV light. So once they're exposed on the plate and the paper, they will fade quite quickly. So in order for you to be able to see the colours, you might need them for identification. The colours will fade, so make sure you take a photo as well straight afterwards. So our hazards, a little bit more hazards for this practical than normal. So we are using very, very fine glass capillary tubing, and they obviously are very easily broken and then can be quite sharp. Using light pressure, you're not pushing or pressing down really hard with them. Make sure they're not rolling off the edges of benches or desks. And if they do break, obviously clear all sharps away appropriately, wash your hands, tell someone that that's happened. The extraction solvent and the running solvent, most of the time you're going to be using something along the lines of it's going to involve propanone, alcohol, methylated spirit. They're going to be very, very flammable and potentially an irritant on your hands. It's a low risk as long as you keep them sealed. So make sure lids are on bottles at all times. Make sure, again, you put it in your vial and you put the bung on, so they're always in sealed containers. They're not just left out open to evaporate, but that you will need them to be out to use them, obviously, and sometimes the lid will need to be off and sometimes they'll be in the pestle and mortar. So if you can do it in a fume hood, great. If not, just make sure the room you're doing it in, the lab, is well ventilated. You've got some windows open, plenty of air space, and the main thing is they're very flammable, so making sure there's no naked flames anywhere near or other sources of ignition. Wearing eye protection while we're doing it in case anything gets in our eye, and obviously standard, if anything gets on your hand, rinse it off straight away. Okay, so here are my 2 leaves. I've got my purple leaf that I said from the plant that's good at protecting itself from UV, and then I've got a very, very bright green spinach leaf as well. So we're going to have a look at trying to compare the pigments in these 2 leaves. Here's my strip of chromatography paper. I'm using just filter paper here, but again, you can obviously use TLC plate as well.
So I'm going to start with the spinach. I'm going to use a pestle and mortar. You can also use a bit of sand to help grind it up. The one thing I would say is just try and avoid using, so no stalks because they're quite tough. And if you split the leaf down the middle, you get rid of that kind of main vein, which is also quite tough as well. So you want maybe 1 or 2 leaves maximum because they're quite big anyway. So I'm going to add some of my solvent to this. Again, not very much. You don't want it to be too liquidy because you do not want it to take too long to dry. Okay, so you can see there I've got a lot of really nice deep green liquid, which I can now use for my chromatography. So I'm going to take a capillary tube, very, very, very fine glass tube. Now we need to work quite quickly because the alcohol, which is the solvent, will begin to evaporate quite quickly. So I'm going to take my pencil and draw a straight line, sort of about a centimetre up from the base of my filter paper. We have to use a pencil because obviously we don't want to use pen because pen can then run. Up along with the pigments. So I've got a very, very small amount of my chloroplast and kind of pigment solution, and I'm just going to dot it gently because I don't want to push all the way through. Onto the middle spot of that line. Now we're going to leave that to dry slightly. You can wave it to dry. You can use a little bit of a hairdryer. There are different ways that you can do this, but then I need to get some more pigment, and I'm going to want to build up that spot. So I've done my green spinach leaves. I'm just going to do the last sort of dot on each one. And I'm going to leave those to dry as best I can now. And I'm going to do my purple leaves now. So I've cleaned out my pestle and mortar. I'm going to need to use fresh capillary tube as well. So these are much finer, but I'm still going to try and remove that middle vein and maybe break them up a bit to help with the process. You can already see that is very perfect.
Okay, so those are my samples done, and now we're ready to actually run the chromatography.
Okay, so I've got my 2 strips. Now the reason I've done 2 of each type of leaf is because what I'm going to try and do is show you the effect that using different running solvents can have. So I'm going to use isopropanol for one set, which is the same solvent that I dissolved the tissue in, and I'm going to use propanone for the other set, and we're just going to see if that affects what the RF values look like at the end and how the pigments are affected and if they look any different, just to show you how important it is to, when you're looking at RF values, that you've used the same running solvent as that value came from when they did their version of that experiment. Okay, so the main things now to be careful of is when we're putting our samples in, I'm going to secure it, but remember our level of our solvent should not come above our pencil line. So it should just be touching the bottom of the chromatography paper when we put it in. And I'm going to secure my paper so it shouldn't be touching the sides, it shouldn't be at an angle, it should just be nice and straight. So I would just check that they fit first, and so that you can kind of gauge where the level is, where you're going to put your solvent. Okay, so now I'm ready to put the solvent in, so I'm going to sort of figure out roughly where I know the bottom of my filter paper is going to be. There we go. So that one is going to start to run. Okay, so I've taken out my piece of filter paper, and I'm just gonna, you don't have to use a ruler, but I'm just gonna use a ruler to draw on the solvent front. So that's where the solvent has reached, the maximum point where it's reached on the paper. And I'm gonna draw that so then we can use those two pencil lines to calculate our RF values.
Okay, so we're going to have a look at trying to analyse our results now and calculating the RF value. Now, as you can see from the picture, my results were a little bit of a smudge. So this can be due to multiple things. It can be due to having too much concentrated on the spot, using the wrong running solvent. A lot of solvents actually take into account different chemicals in the pigments that can actually interfere with the chromatography, and you sometimes need a mixed solvent to be able to get a really good achieved separation. And so that is what I've got here. This is one that I did on a thin layer chromatography plate using spinach again, so it's the same plant. And so I've measured my solvent distance, which is 36 millimeters. So now I can start calculating the RF values for all of those really nice separate distinct points, and then we can use their colors as well as their position and their RF value to try and work out what pigments we've got separated in these leaves. So the first point is just up from the baseline. So that was measured, the distance from the baseline to the middle of the spot was 5 millimeters. So using our formula, 5 divided by 36 millimeters, which was the distance that the solvent has traveled, gives us an RF value of 0.14. Okay, so I'm going to go ahead and repeat all of those calculations for the rest of the spots. Make sure to measure to the middle of those spots each time. Okay, so now I've got all my RF values, I can start trying to identify the spots. The nice big bright yellow spot right at the top, the 6 spots, clearly going to be a carotene because it's yellow and orange, a bright yellow, deep yellow, and it's right at the top and it's a 0.9 RF value, which fits right in. The phytin is quite easy to spot because it's grey and there aren't any other grey spots, and it will be responsible for that, even if the RF value isn't exactly the same. Then we've got chlorophyll A normally comes before chlorophyll B if we're going kind of down from the top. And we've got the blue-green, and you can see we've got a blue-green spot and then we've got like a sort of bright light green spot. So we've got 2 greens sort of close to each other and we've got that blue-green. So I think that blue-green is most likely going to be chlorophyll A. And then obviously chlorophyll B underneath, and it could be a couple of chlorophylls. There are more than just the 2 chlorophylls as well, so there can be some other chlorophylls in there. Now I'd say we probably don't really have any anthocyanins at all because there's no purple, but we'll look at what that looks like in a second when I show you my chromatogram that I got from my purple leaves. Xanthophylls, we could have some xanthophylls, that yellow-green, that very, very early kind of yellowy-browny spot we could have down the bottom. It could be a xanthophyle, because if you look at it, our RF value was about 0.14, and 0.15 has been shown to be an RF value for xanthophylls. Bear in mind, obviously, we can't necessarily use these RF values and directly match them up. These three different columns are all from RF values that were done with completely different running solvents in completely different conditions. So they will never be exactly the same unless you run it in exactly the same way. Okay, so I said there were no xanthophylls present on that chromatogram we just measured, but there were some present on mine. So you can see where I had my purple leaves, I've got very, very clear purple runs that have gone up, and you can see how they've started to separate, and then they've gone into green. So there's two interesting things here, is that we can see what anthocyanins look like on a chromatogram, and we can see that they're there, and they do sort of separate out quite early on down the bottom, which is as expected. And also we can see the difference between the two running solvents. So one very clearly separated out this green and the purples as two sections, whereas the other one, there was only a little bit of green here. It clearly travelled a lot slower, and it wasn't necessarily going to spread it out as far. But it's also very nice to show that those very purple leaves did have obviously chlorophylls present, because that's what gives us the green colours, and obviously it has to have those because it will have to have those in its chloroplast to be absorbing the wavelengths of light that we need for photosynthesis. And most of those other coloured pigments, sort of the purples, the reds, are those accessory pigments that can help to get extra wavelengths. So hopefully that was helpful. That is sort of tips and ways and methods for carrying out this chromatography practical that you're going to do as part of the photosynthesis topic.
Ouch! This is why in some videos I have unexplained scratches.